Hello everyone and welcome to the new session of the lecture series, and today's topic is cell cycle and cell division. And today we'll be discussing and solving all the NEET previous year questions. So let's start with the first question. Here they are asking the process of appearance of the recombination nodules, okay, occurs at which substage of prophase 1. Okay, you can do one thing, you can underline all the keywords in the question and then you can concentrate on that. Third keyword is the meiosis. Okay, so recombination nodules, help in the recombination and the crossing over. All right, so what should be the answer? Answer should be the Peckettine stage of prophase 1 of meiosis. All right, so meiosis, okay, is divided into two phases, meiosis 1 and we have the meiosis 2. Here we have the interphase and then we have the prophase. Prophase is divided into leptotene, zygotene, pectine, diplotene and diakinesis. Okay, so the recombination nodules are formed during the pectine stage of the prophase. These recombination nodules helps in the crossing over, or you can say exchange of genetic material. Now second question is, the dissolution of synaptonymal complex occurs during, okay, synaptonymal complex they are asking. Alright, so initially what happens? Again, let's talk about the substages of the prophase. Okay, we have leptotene, we have zygotene, pectine, we have diplotene, and then we have diakinesis. Okay, so synapses formed during which stage? Zygotene. Okay, or you can say synaptonymal complex also forms during zygotene. Alright, so zygotene can't be the answer. They are saying the dissolution of synaptonymal complex, not the formation. Okay, so don't get confused. In pectine, what happens? Crossing over takes place. Okay, so in diplotene, what happens? Chiasmata formation is seen and the homologous chromosomes separate. Okay, they start separating. In diakinesis, terminalization of chiasmata is seen. Terminalization of chiasmata or chiasma is seen. Okay, so what should be the answer? Dissolution of synaptonymal complex leads to the, we already know, separation of the homologous chromosome. Okay, so answer should be diplotene. Alright, now coming to the third question, here they are asking that doubling of the number of chromosome can be achieved by disrupting the mitotic cell division soon after. Okay, options are anaphase, telophase, prophase and metaphase. Okay, doubling of the number of chromosome they are asking. So what should be the answer? Answer is the metaphase stage. Alright, they are asking doubling of the chromosome, okay, during which stage of mitosis takes, okay. So in metaphase, what happens? All the chromosomes are lined at the equator. Okay, all the chromosomes are aligned at the equator, or you can say equatorial plate or the metaphase plate. Okay, so what happens? During anaphase, what happens? Okay, there is, in mitosis, there is separation of the chromatids. Alright, so if we block the separation of the chromatids, okay, during the anaphase, then we will achieve the doubling of the number of the chromosome. So if we disrupt the mitotic cell division during the metaphase, then we will get the doubling of the chromosome and chromosome will not separate during the anaphase. Now, let's come to the fourth question. During which stages of mitosis and meiosis, respectively, does the centromere of each chromosome split? So they are asking at which stage the centromere splits. Okay, so centromere splits and sister chromatids separate because of the splitting of the centromere. If the centromere will not split, then chromosomes will separate and this will lead to the reductional division. Okay, reduction of the number of chromosome. So metaphase we know, chromosome aligning at the equator. Alright, so this cannot be the answer. Prophase and telophase they are saying. Okay, prophase, condensation of chromosome is taking place and all everything, nucleolus, nuclear membrane is disappearing. Telophase, what is happening? It is the reverse of prophase. Alright, so reverse of prophase is happening, appearance of nuclear membrane and nucleolus and the cell organelles. Alright, so this cannot be the answer. Now they are saying telophase and anaphase 1. Again, telophase cannot be the answer. Anaphase 1 they are saying. Okay, so what happens during the anaphase 1? Separation of chromosomes takes place. Okay, and not chromatids, leading to, this leads to the reductional division, or you can say reduction of the chromosomes. Alright, so what should be the answer? We have this option left, anaphase and the anaphase 2. So anaphase, they are here, they are talking about the mitotic anaphase. Okay. Here there is separation of chromatids due to the splitting of the centromere. Okay, centromere splits here. Centromere splitting. Here also, anaphase 2 also, what happens? There is centromere splitting and separation of chromatids leading to the equational division. Okay, that's why mitosis is known as equational division. Anaphase 2 is also known as the equational division. It is similar to mitosis. So answer is D. Now coming to the fifth question, which one of the following is the quiescent stage of the cell cycle? Okay, so they are talking about the interphase. So interphase we know is divided into G1, S and G2 phase, and then we have the mitotic phase. Alright. So what happens? Some of the cells such as heart cells, nerve cells, skeletal muscle cells or some lymphocytes, they exit the G1 phase. Alright, and they enter a G0 phase, which is known as the quiescent phase or stage, or you can say it is an inactive stage. Okay, so what should be the answer? Answer is the G0 phase. It is also known as the quiescent stage or the inactive stage. And depending on the condition, the cell can re-enter the cell cycle. It can re-enter the G1 phase. Now coming to the sixth question, which of the following stages of meiosis involves division of the centromere? Again, it is a similar question to the previous one. So what should be the answer? Metaphase 2, chromosomes are aligned at the equator. Telophase, which is the reverse of prophase. Alright. It is the reverse of prophase. Alright. Anaphase 2 we have. Anaphase 2, what happens? Splitting of chromosome takes place here. Splitting of centromere. Not chromosome. Splitting of centromere takes place here, leading to the separation of the chromatids. Metaphase again, chromosomes are lying at the equator. So what should be the answer? Answer should be anaphase 2. Alright, anaphase 2 of meiosis. Now coming to the next question, here they are asking, among eukaryotes, replication of DNA takes place in, alright, again they are talking about the interphase here. So interphase again divided into G1, S and the G2 phase. Okay, G1 what happens? There is cell growth is taking place. Cell is metabolically active. There is production of macromolecules like carbohydrates, proteins, lipids, RNA. S phase what happens? There is synthesis of DNA, or you can say DNA replication takes place here. G2 what happens? In G2 phase, again there is final preparation for cell division is taking place. Final preparation for the cell division. And again there is synthesis of some organelles also, or you can say cytoplasmic organelles also, they double, like endoplasmic reticulum, Golgi complex and all. So what should be the answer? They are asking here the replication of DNA. M phase. Okay, what M phase? After G2 phase, we have the M phase, which is the actual cell division phase. Alright. M phase, what happens? Is further divided into karyokinesis, which is known as the nuclear division, and the cytokinesis, which is known as the cytoplasmic division. Okay, so the answer should be the S phase. DNA replication takes place during the S phase of the interphase. So let's start with the next question, which is, here we have to match list with list 2. Okay, so now M phase. Okay, so here they're talking about the mitotic phase or the mitosis. Okay, now it is related to equational division. Okay, now G2 phase. G2 phase, what happens? Protein synthesis takes place during G2 phase. Alright, then next is the quiescent stage. Okay, so quiescent stage is an inactive phase. Okay, when the cells exit the G1 and they enter the G0 phase. Okay, also known as the quiescent stage. Now next is G1 phase. During G1 phase, what happens? So let's again study interphase. Interphase is divided into G1 phase, S phase, G2 phase, and then we have the M phase, mitotic phase, or sometimes meiosis phase also during meiosis. So G2 phase, they are saying protein synthesis there takes place. Okay, and the cell, what is doing? It is preparing for the cell division. The final preparation of cell division is going on here. During S, what happens? DNA synthesis or DNA replication takes place here. G1 again, what happens? Cell growth is taking place. Okay. M phase, mitotic phase, is known as the equational division. Okay, if the cell is parent cell was diploid, it will give rise to two genetically similar, okay, daughter cells with the same chromosome number. Okay, so here A is 4th, then B, G2 should be 1, then C, quiescent stage, 2nd, inactive phase, and then D is G1, interval between mitosis and the initiation of DNA replication. G1 is interval between mitosis and initiation of DNA replication. So G1 is, which is D, is 3rd. So these are the options. Okay, you're going to have a look at the options. So what should be the answer? Answer is option B. Now moving on to the next question. Now here we have to select the correct statement. First is tetrad formation is seen during leptotene, they are saying. Second is during anaphase, the centromere splits and the chromatid separate. Okay, third is terminalization takes place during pectine. Fourth option is nucleolus, Golgi complex and ER are reformed during telophase. Fifth option is crossing over takes place between sister chromatid of the homologous chromosome. Okay, so tetrad formation they are saying is seen during leptotene, which is not true. Tetrad formation is seen during zygotene, because during zygotene bivalents are formed. Bivalents are formed, okay, during the zygotene and not leptotene. Bivalents are also known as tetrads because they contain 4 chromatids. Okay, if this is a bivalent, means pairing of homologous chromosome forms a bivalent. First chromatid, second chromatid, third chromatid and fourth chromatid. So it is a tetrad. Now they are saying during anaphase, the centromere split and the chromatid separate. Alright, this we already know. Centromere splitting takes place during anaphase and the chromatid separate during the anaphase of mitosis and the meiosis too. So this is correct. Terminalization takes place during the pectine stage. Alright, which is again not true, because terminalization takes place during the last stage, diakinesis. Terminalization of chiasmata they are saying. Okay, terminalization of chiasmata. Now, nucleolus, Golgi complex and ER are reformed during telophase. Alright, which is again true or not? So this is true. Okay, because telophase is reverse of prophase. Okay, in prophase, during prophase, what happens? Nucleolus, Golgi complex, ER, as well as nuclear envelope, they all disappear. But during the telophase, they reappear. That's why telophase is known as reverse of the prophase. Okay, so this is also true. Now, coming to the last option, crossing over takes place between sister chromatid, it is saying. Okay, and this is also not true because crossing over takes place between non-sister chromatid. Okay, crossing over always takes place between the non-sister chromatid. So if this is a pair of homologous chromosome, okay, crossing over takes place between these two. These are known as the non-sister chromatid. And these are the sister chromatid. Alright, so what should be the answer? These are the options: B and D, A, C, D, B and E, and A and C. So what should be the answer? Answer should be B and D. Now next question is, given below are the two statements. Here they are giving the two statements, statement 1 and statement 2. So you can see here there is so much diversity in the questions. Okay, there are within the MCQs also. Okay, there are so much diversity in the questions. So this is a statement type of questions. So what is the first statement? During the G0 phase of the cell cycle, the cell is metabolically inactive, it is saying. Alright. Statement 2, they are saying the centrosome undergoes duplication during the S phase of the interphase. Alright. So now what they are asking? Okay. In the light of the above statement, choose the most appropriate answer from the options given below. First is both statement 1 and statement 2 are incorrect. Okay. Statement 1 is correct, but statement 2 is incorrect. Third is statement 1 is incorrect, but the statement 2 is correct. Fourth is statement 1 and statement 2 are both are correct. Alright, so what should be the answer? So here they are saying metabolically inactive. But actually G0 phase is metabolically active. Alright, but the cells do not proliferate. Means they are not dividing. But the cell is actually active. So this is false statement. It is not true. Whereas the centromere undergoes duplication during S phase. Alright, so this is true. Along with the DNA synthesis, centrosome also undergoes duplication. What is a centrosome? Centrosome is a structure within which microtubules are present, which consist of microtubules which are involved in spindle formation. Alright. Or you can also say centrosome help in the organization of the microtubules, or you can say spindle fibers. So what should be the answer? Statement 1 is incorrect, whereas statement 2 is correct. Now coming to the next question. Here they are asking which stage of meiosis can last for months or years in the oocytes of some vertebrates. Okay, what should be the answer? Options are dikinesis, leptotene, pectine and diplotene. So the answer is diplotene. Okay, so sometimes what happens, meiosis, known as the reductional division, can last for months or even years in the oocyte. Now what is an oocyte? Oocyte is a developing egg. Okay, so sometimes the meiosis gets stuck in the diplotene stage. Okay, and what are some of the reasons? Okay, there are some of the reasons has been given. Some of the reasons are, first is the chromosomal pairing. Okay, so if the meiosis is stuck at the diplotene stage, it helps the chromosome to pair, okay, very efficiently. Okay, so it helps in the pairing of the chromosome. If there is no pairing of the chromosome, then later on there will be problem in separation of the chromosome. So it gives ample time for the chromosomes to get paired nicely. Okay. Now, some of the other reasons are there. Okay, sometimes what happens, the chromosomes, or you can say DNA. Okay, DNA is not being replicated properly, or there is damage in the DNA. Okay, so this, okay, gives adequate time for the cell to repair the DNA. okay, for repairing of the DNA. Okay, so that's why sometimes the meiosis stage, okay, gets stuck in the diplotin stage in the oocytes of some of the vertebrates. All right, there is one more, you can say one more reason for the meiosis to get stuck in the diplotin, that is the regulatory mechanism. Okay, third you can say is the regulatory mechanism. Regulatory mechanism means it gives time for the cell to regulate the cell cycle. Okay, so these are the three probable reasons why the meiosis gets stuck in the diplotin stage of. Okay, now coming to the next question. Here we have to identify the correct sequence of events during prophase 1 of meiosis. Alright, prophase 1 is further divided into leptotin, zygotin, pectin, diplotin, and diakinesis. Alright, so first they are asking synapses of homologous chromosome. This takes place during zygotin. Chromosome become gradually visible under microscope. Leptotin. Crossing over between non-sister chromatids, okay, takes place during pectin. Then we have terminalization of chiasmata, takes place during the last stage, diakinesis. Dissolution of synaptonymal complex takes place during diplotin. So what should be the answer? These are the options. Okay, what should be the answer? Answer is D. Okay, let's again check our answer. Here they are saying D, B, A, C, E, and D. Alright, so first what is happening? Chromosome become gradually visible under the microscope. So first stage is leptotin. Okay, then we have synapses of homologous chromosome. Okay, then what will happen after synapses? Crossing over will take place. After crossing over, dissolution of synaptonymal complex, and finally we have terminalization of chiasmata. So this is the answer. Now coming to the next question, here they are saying bivalent or tetrad formation is a characteristic feature observed during which stage? Okay, so here they are again talking about the prophase 1 of meiosis. Alright, so what should be the answer? Bivalent are seen during the, here, chiasmata in zygotin they are saying. This is not true. Okay, synaptonymal complex during zygotin stage, again not true. Or true? Oh, here they are saying synaptonymal complex in the zygotin stage. Alright, so this is true. Chiasmata in diplotin stage, not true. And synaptonymal complex during pectin stage. This is not true. So what should be the answer? Answer should be synaptonymal complex in the zygotin stage. Let's talk about the next question. Here they are saying with respect to metaphase, which of the following statement is incorrect? Okay, first is chromosome lie at the equator. This is true. Chromosome disintegration, complete disintegration of nuclear envelope. Okay, true. Chromosomes are highly condensed. True. Metaphase chromosomes are made up of 4 sister chromatids held together by the centromere. Okay, so what we have to here identify the incorrect statement. So answer should be D. Okay, metaphase chromosomes are made up of 4 sister chromatids held together by the centromere. This is incorrect statement. Okay, metaphase chromosome made up of 2 sister chromatids. held together by the centromere. Now let's talk about the next question, which is here they are asking the appearance of recombination nodules on homologous chromosome during meiosis characterizes. First one is synaptonemal complex, then we have the bivalent, then sites at which crossing over takes place, and terminalization. So what should be the answer? Recombination nodules, okay, are the area or the site where crossing over takes place. Okay, where crossing over takes place. So answer should be C. Okay, answer is C. They are the sites at which crossing over occurs. Now, next question is here we have to again match list with the match list 2. Alright. So here they are talking about the types of chromosome. Okay, first is the metacentric chromosome, acrocentric chromosome, submetacentric, and then we have the telocentric chromosome. Alright. So these are the options. Okay. So first let's talk about the type of chromosome. First is metacentric chromosome. Okay, where the centromere is present in the center. Based on the position of the centromere, the chromosome has been classified. Okay. So if the centromere is present in the center, it is known as the metacentric chromosome. Okay. Metacentric chromosome. Then what is submetacentric? Here the centromere is present just near to the centromere. Okay. Or you can say just above the centromere. This is known as submetacentric. Submetacentric. And then we have acrocentric. Okay, where the centromere is towards one end of the chromosome. Okay. So here one arm is very short, other arm is longer. Okay. Here the centromere is present here. Okay, towards one of the end. Okay, this is acrocentric. And then we have the last one, which is the telocentric. Here the centromere is at the terminal position. Okay. This is telocentric. Position of the centromere is terminal. Okay. Now, so what should be the answer? Answer is first one. Alright. Metacentric, centromeres in the middle forming two equal arms. What is acrocentric? Okay. What should be the acrocentric? Acrocentric is centromere situated close to the end forming one extremely short and one extremely long arms of the chromosome. Submetacentric, centromere slightly away from the middle forming one shorter arm and one longer arm. Okay. And telocentric, centromere at the terminal end. Alright. So they are talking about the one short arm and one long arm. Okay. This is the short arm of the chromosome. This is the long arm of the chromosome. Now coming to the next question. Here they are saying regarding meiosis, which of the statement is incorrect? Okay, we have to identify the incorrect statement. First is there are two stages in meiosis, meiosis 1 and meiosis 2. This is correct. DNA replication occur in S phase of meiosis 2. Alright, they are talking about meiosis 2. So this is incorrect. The DNA replication takes place during meiosis 1 and that too during the interphase. Okay, interphase you know is divided into G1, S and G2 phase. DNA replication or DNA synthesis takes place during the S phase. Pairing of homologous chromosome, this is true. Okay, which is known as synapsis. This occurs during the zygotine stage. 4 haploid cells are formed at the end of meiosis 2. This is also true. So what should be the answer? DNA replication occurs in the S phase. This statement is incorrect. Now, coming to the next question, here again we have to select the incorrect statement with reference to mitosis. Okay, mitosis, okay. The earlier question was meiosis. All the chromosomes lie at the equator at metaphase. True. Spindle fiber attached to centromere of the chromosome. Okay. So this is true or not? We will see. Chromosome D contains at telophase. This is also true. Splitting of centromere occurs at anaphase. This is also true. Okay. So what should be the answer? Answer is B. Okay. So this is half correct answer. Spindle fiber attached to the centromere, but they at which area? They attach to the kinetochore of the centromere. So this was a bit tricky. Alright. So here you have to say the spindle fiber attached to the kinetochore, which are the areas present on the centromere. If this is the chromosome, two kinetochores are present like this, okay? And here we have the chromosome. These are kinetochore, which are areas present on the centromere. Now, next question is, which of the following stages of meiosis involve division of the centromere? This is again the similar question. We have already done this type of question. Telophase 2, metaphase 1. No, the chromosomes are aligned at the equator. Alright. Metaphase 2, again chromosomes are aligned at the equator. The answer should be anaphase 2. Now, next question is the complex formed by a pair of synapsed homologous chromosome. Okay, options are bivalent, exoneme, equatorial plate, or kinetochore. So this is quite simple. The answer should be bivalent. Okay, the pairing of synapsed homologous chromosome, okay, forms a bivalent, also known as tetrad. Okay, now let's talk about the next question, which is, here we have to, okay, here a figure is given. So this is another type of question, okay. Here we have been given a figure, and what we have to do, we have to identify this image. Okay, we have to identify the image here. So this was the end of the session, and we will be discussing about rest of the question in the next session. Thank you very much.